Okay, let's dive right into something really, really fascinating today. Imagine these tiny, sort of invisible organisms locked in these ancient battles,
Hmm.
but not with armies, right there inside the cell itself.
It sounds pretty wild, doesn't it? But yeah, this isn't sci-fi. We're talking about bacteria and archaea and this really complex system they have called CRISPR.
CRISPR, yeah, that's a word you hear constantly these days. Usually it's tied to gene editing, right? But its original job, its natural role, is totally different.
It really is. Fundamentally, CRISPR is it's a type of acquired immunity, and it's heritable.
Acquired and heritable.
Yeah, it's this really sophisticated defense system these microbes use to fight off invaders. Specifically viruses like bacteriophages that attack them and other foreign bits of DNA like plasmids.
So wait, a microscopic immune system that actually remembers past infections? That seems advanced.
It is, and this system, this bacterial defense mechanism, turned out to be the foundation for that whole gene editing revolution.
Yeah.
The one that won the Nobel Prize even.
Amazing. It's just incredible how that works.
It really is. A perfect example of how, looking deep into the basic biology of what seem like simple organisms can lead to these massive breakthroughs, things that change medicine, everything.
And that's exactly what we're doing today. We've dug into the sources to really unpack this whole story, how CRISPR was discovered, how it actually works, like at the molecular level, some of the unexpected twists.
Oh, yeah, they are twists.
And then, yeah, touching on those huge applications. It apparently all started with some, well, kind of accidental observations.
It really did. It wasn't like someone set out to find an immune system. The first sort of hint came way back in 1987. Yoshizumi Ishino and his team in Osaka.
Okay.
They were studying a gene in E. coli, just regular lab work, and they noticed these weird repeated DNA sequences.
Weird how, I mean, DNA is full of repeats, isn't it?
Yeah, but these were different. They were interspersed with other unique sequences, like repeat spacer, repeat spacer. They noted them down, published it, but honestly had no clue what they did. Just a curiosity.
It's just a weird pattern. So what happened then? Did others see them too?
Yep. Similar structures started popping up elsewhere. In '93, a group working on Mycobacterium tuberculosis, the TB bacterium, they noticed that the sequences between the repeats, the spacers, were super variable between different strains.
Ah, so they could use it to tell strains apart.
Exactly. They developed a typing method based on it called spoligotyping. It's actually still used today. So useful, even if they didn't know the why.
Right, a practical tool from a mystery.
Mm-hmm. And around the same time, this researcher in Spain, Francisco Mojica, was really digging into these repeats, initially in archaea. He was the first one to really characterize them systematically. He found they were even transcribed into RNA.
Okay, so getting closer to function, maybe.
Getting warmer. By 2000, his group had scanned a bunch of genomes and realized, hey, these repeat clusters are everywhere. Like in 20 different microbes at that point, a whole family of structures.
Did he call them CRISPR back then?
Not quite yet. His first name was SRSR short, regularly spaced repeats. Bit of a mouthful.
Yeah.
The name CRISPR, clustered regularly interspaced short palindromic repeats, that came about in 2001. It was a collaboration between Mojica and Ruud Janssen in the Netherlands, who was also tracking these sequences.
And Janssen, he made another important connection, didn't he?
He really did. Janssen spotted that these CRISPR repeats were almost always located near a specific set of genes. He called them Cas genes for CRISPR-associated systems.
Has genes.
And looking at what those Cas proteins look like structurally, they had bits suggesting they could, mess with DNA or RNA, like cut them or unwind them. That was a big clue that this wasn't just static DNA, it was part of some active process.
Okay, so the plot thickens. We've got the weird repeats, the variable spacers, the associated Cas genes that look like they do something, but still no definite function. This must have been the point where people really started chasing it.
Absolutely. The massive breakthrough, the real aha moment came in 2005. And it wasn't just one group, it was 3 independent teams, including Mojica's again.
What did they find?
Yeah. They all converged on the same stunning discovery. Those unique spacer sequences, the bits between the repeats. They matched sequences from foreign DNA.
Foreign DNA? Like what?
Like viruses, bacteriophages that infect those bacteria, or from plasmids, those little extra circles of DNA bacteria can swap.
Oh, wait. So the bacteria were literally taking snippets of their attacker's DNA
Yeah.
and storing them in their own genome, like a most wanted gallery.
That was exactly the incredible implication. It screamed adaptive immunity. a way for bacteria to remember past infections and recognize them if they showed up again.
That is groundbreaking for bacteria.
Totally. And here's a wild bit of history. Apparently, the initial papers reporting this finding, they got rejected from some top journals.
Rejected? Why?
The idea of adaptive immunity in bacteria, it was just too radical, too unexpected back then. People weren't ready for it, I guess.
Wow. Science can be tough.
Yeah.
So how did they finally prove it experimentally, that it really was adaptive immunity?
That solid proof came in 2007 from Rodolphe Barangu and his team. They were working at Danisco, a food ingredients company, actually.
Yeah.
They were studying Streptococcus thermophilus. That's one of the bacteria used to make yogurt and cheese. And they were having problems with phages killing their cultures.
Ah, practical problem.
Exactly. And they did these really elegant experiments. They deliberately exposed their S. thermophilus cultures to specific phages, the bacteria that survived the attack. They checked their CRISPR arrays, and they found the survivors had incorporated new spacers into their CRISPR locus. And guess what?
They matched the phages they'd just fought off.
Perfectly. And then they went further. They engineered bacteria, deliberately adding or removing specific spacers, and they showed that adding a spacer against a phage made the bacteria resistant to that phage. Removing it made them susceptible again.
That nails it. Direct cause and effect.
Absolutely. That was the first undeniable experimental proof. CRISPR is an acquired adaptive immune system against viruses, found essentially because of yogurt.
Yogurt bacteria? Who knew? Okay, so now we know what it is. An adaptive immune system. How does it actually work? How does it pull off this trick of remembering and then fighting?
Right. So it basically operates in three main stages, all designed to find and destroy foreign nucleic acids, DNA, or sometimes RNA.
3 stages. Okay, stage 1.
Stage 1 is spacer acquisition. You can think of this as the learning phase, or building the memory. When a bacterium gets infected for the first time by a new virus, say, it needs to capture a piece of that invader's DNA. Little identification tag. We call that piece the protospacer.
Protospacer. How does it actually grab that piece? Does it just snip randomly?
Not quite randomly. It uses specific proteins, the Cas proteins, particularly Cas1 and Cas2. These two are found in almost all CRISPR systems, and they work together, forming a complex.
Cas1 and Cas2. And they do the snipping and inserting?
Exactly. They act like molecular scissors and glue. They cut out a fragment, the protospacer, from the invader's DNA, and then they integrate it, paste it into the bacterium's own CRISPR locus in its chromosome.
And where in the CRISPR locus do they stick this new memory?
Typically, new spacers get added at one specific end of the CRISPR array, right near a sequence called the leader sequence.
The leader sequence.
Yeah. And because they're always added at that end, it creates this amazing chronological record. The spacers closest to the leader are the newest infections. The ones further down are older memories.
Like a little timeline of past battles fought?
Pretty much, yeah, a genetic history book of infections. Now, this capture process isn't totally random in terms of what gets captured either. There's another key signal involved, especially in the type I and type 2 systems.
Okay.
It's a very short DNA sequence on the invader's DNA, right next to the protospacer sequence that gets grabbed. It's called the PAM, protospacer adjacent motif.
PAM, what does the PAM do?
It's crucial. It acts like a little grab here sign for the Cas1, Cas2 machinery. It helps them identify a suitable protospacer on the foreign DNA and influences exactly where they cut and how the new spacer gets inserted nice and tidily into the CRISPR array. It ensures specificity.
So the PAM guides the whole acquisition process, makes it accurate.
Exactly. And there's this other cool feature called priming, or primed acquisition.
Priming.
Yeah. If the system encounters foreign DNA that's similar but maybe not a perfect match to a spacer it already possesses, it actually becomes much better and faster at acquiring new spacers from that same invader DNA molecule. It's like the system says, I recognize this guy. Let's learn more about him quickly.
So prior exposure boosts the learning process. That's
Right.
pretty sophisticated for bacteria. Okay, stage 1, grab the invader's ID card, guided by PAM, and store it. Got it. Stage 2.
Stage 2 is called biogenesis, or more accurately, cRNA biogenesis. This is about making the actual wanted posters from those stored memories.
Wanted posters. I like that. How?
The cell transcribes the entire CRISPR locus, all the repeats and all the spacers, into one single long RNA molecule, a pre-cRNA.
The whole history book gets copied into RNA.
Yep. But that long molecule isn't useful yet. So other Cas proteins step in and process it. They chop it up into smaller functional units. Each unit is a mature CRISPR RNA, or RNA.
Each Cas RNA.
Each mature kRNA contains the sequence from one spacer the invader signature flanked by bits of the repeat sequence from the CRISPR array.
So that chRNA is the actual guide molecule that will hunt down the invaders.
Exactly. That chRNA is the key guide. Now, how this processing happens, how the long RNA gets chopped up, it actually varies a bit between the different CRISPR types.
Oh.
Yeah, like in type I and type 3 systems, they often use an enzyme called Cas6 that recognizes the structure of the repeat sequences in the RNA. But in type 2 systems, the famous Cas9 ones, it's a bit different. It involves another small RNA called the tracRNA transactivating chRNA, which pairs up with the repeats, and then a standard bacterial enzyme called RNase III does the cutting, along with Cas9 itself playing a role.
Okay, so different methods, but the end result is the same. These small cRNA guides, each carrying the fingerprint of a past invader.
Precisely. Which brings us to stage 3, interference. This is the action phase, the defense itself.
Okay, the attack.
Right. The mature cRNA doesn't work alone. It teams up with one or more Cas proteins to form an interference complex, sometimes called effector complexes, like the cascade complex in type I systems.
So the guide RNA joins forces with the Cas enforcer proteins. What's their mission?
Their job is to patrol the inside of the cell. They're constantly scanning, searching. The cRNA molecule within the complex acts as the guide, using its spacer sequence to look for matching sequences in any DNA or RNA floating around.
Searching for a match to the stored memory, like facial recognition for DNA?
Kinda, yeah. And here's where the PAM comes back in, at least for type I and type 2 systems targeting DNA. Finding the matching sequence based on the cRNA guide isn't quite enough.
The PAM again.
Yep. For these systems to launch the attack, the interference complex has to recognize not only the target sequence matching the cRNA, but also the correct PAM sequence right next to it on the invading DNA.
So the PAM acts like a necessary confirmation signal. Yes, this is definitely the bad guy. Proceed.
Exactly. It's a crucial checkpoint. It prevents the system from attacking the wrong things. Once both the target match and the PAM are recognized, then the Cas proteins in the complex get the green light.
And they cut.
They cut. Proteins like Cas3 in type I systems, which shreds the DNA, or the famous Cas9 in type 2 systems, which makes a clean double-strand break in the DNA. Target neutralized.
Efficient. You mentioned type 3 is different, though.
Right. Type 3 systems are fascinating. They can target DNA, but they can also target mRNA, the messenger RNA copies of genes. This lets them fight off RNA viruses, potentially.
Okay, RNA targeting. Do they need a PAM for that?
Here's a key thing. They typically don't need a PAM to target and cut RNA. They have a different mechanism for recognizing the target RNA.
More versatile, then. But here's a question that seems obvious. The bacterium has the CRISPR locus in its own DNA. That locus contains the spacer sequences, right? So how does the system stop itself from attacking its own chromosome? How does it know not to cut its own memory bank?
That's a brilliant and crucial question. It's about self versus non-self discrimination. The answer lies partly in those bits of repeat sequence that are included on the cRNA, flanking the spacer.
The repeat bits on the guide RNA.
Yeah. If the RNA tries to bind back to the CRISPR locus itself, it will match the spacer sequence, but it will also match the repeat sequences very well. This extensive pairing, especially to the repeat part, acts as a signal. It essentially tells the Cas complex, hold on, this looks like self, don't cut here.
So the guide RNA binding too perfectly, including the repeat parts, prevents the attack.
In essence, yes. The lack of a PAM sequence within the CRISPR locus itself is also critical for systems like type 2. But that interaction between the qRNA's repeat portion and the host DNA is a key abort attack signal. It's very elegant.
It really is. Okay, so we've mentioned type I, 2, 3. It sounds like there's quite a variety of these systems out there.
Oh, absolutely. There's huge diversity. Scientists classify them into two broad classes, class 1 and class 2, and then multiple types like I, 2, 3, 4, V6, and even subtypes. They differ mainly in the collection of Cas proteins they use.
So the core idea is similar, but the toolkit varies.
Exactly. Class 1 systems, like type I and 3, tend to use multi-protein complexes for interference. Class 2 systems are simpler, often relying on just a single large protein, like Cas9 or Cas12.
Are there other really notable types besides I, 2, and 3? You mentioned Cas9 is the gene editing star.
Definitely. Besides type 2 with Cas9, there's type V. This includes proteins like Cas12a, sometimes called CPF1. It's also a DNA cutter like Cas9, but it's different.
How so?
Cas12a makes staggered cuts in DNA, not blunt cuts like Cas9. It recognizes a different PAM sequence, usually T-rich. And interestingly, it processes its own cRNAs and only needs the cRNA, not the separate tracRNA that Cas9 needs. So it's another great tool for gene editing, just with slightly different properties.
Another option in the toolbox. Cool. Any others?
Yeah, type 6 is really interesting. These systems use Cas13 proteins, and Cas13 targets single-stranded RNA, not DNA.
RNA targeting again, like type 3.
Yes, but Cas13 has this peculiar feature called collateral cleavage. Once it finds and binds its target RNA,
Uh-huh.
it goes kind of hyperactive and just starts shredding other RNA molecules nearby, non-specifically.
Oh, like a sort of RNA bomb goes off?
Kind of. It's a bit destructive, but this very property has been cleverly harnessed. Scientists developed diagnostic tools based on it. If the target RNA, say from a virus, is present, Cas13 activates, shreds reporter RNAs, and creates a detectable signal.
Wow, turning a weird side effect into a diagnostic test? That's clever. So
Yeah.
DNA editors like Cas9 and Cas12, RNA editors like Cas13, it really is a versatile natural toolkit.
It really is. And thinking about all this diversity points to a long and dynamic evolutionary history.
Yeah, it must be a real arms race going on.
Totally. It's classic coevolution. Bacteria and archaea versus their phages. The case genes themselves probably evolved from bits and pieces of other systems,
Yeah.
maybe mobile genetic elements, ancient stuff. But once CRISPR-Cas immunity emerged,
Your phages had to respond.
exactly,
Yeah.
relentlessly, it's this constant back and forth. Bacterium acquires a new spacer against a phage. The phage population evolves mutations, maybe changing the sequence the spacer targets, or crucially mutating the PAM sequence so the Cas proteins can't recognize it anymore.
So the phage learns to evade the bacterial wanted poster.
Right. Molecular hide and seek, constantly escalating. And this whole dynamic, it brings up this really fascinating kind of philosophical debate about Lamarckian evolution.
Lamarckian. You mean like inheritance of acquired characteristics? I thought that was disproven.
Well, mostly. Yes, for complex organisms. But CRISPR is interesting. The Cas proteins evolve through standard Darwinian, means random mutation, natural selection. But think about the spacers. The bacterium acquires that spacer DNA directly from its environment, the invading virus, in direct response to an attack. That acquired piece of information is then integrated into the genome and passed down to its offspring.
So the offspring inherit resistance that the parent acquired during its lifetime.
Precisely. Which some people argue fits the definition of Lamarckian inheritance, at least in this specific context. It's nuanced, of course, but it's a really provocative idea that challenges how we usually think about evolution.
An environmentally acquired trait being directly inherited.
Yeah,
That's
but-
mind-bending. Do we see this evolution happening fast in nature? Like, can we watch bacteria pick up new spacers?
Oh yes, it depends hugely on the environment.
Yeah.
Those Streptococcus thermophilus studies in dairy cultures, where they're under constant attack by phages,
Yeah.
they showed new spacers being acquired incredibly rapidly, sometimes within days or weeks of a new phage challenge. Really fast adaptation.
High threat level, high speed evolution.
Exactly. But then you look at other bacteria, maybe like E. coli in a more stable environment, or Salmonella, and their CRISPR arrays can be stable for very long periods. The rate of change seems really tuned to the level of threat they face. Studies looking at microbes in all sorts of places, from acidic mine runoff to inside our mouths, show this huge variation in CRISPR dynamics.
Makes sense. The defense system ramps up when the war is hot. Okay, you mentioned earlier there were some unexpected twists in the CRISPR story. What kind of surprises came up?
Well, one of the biggest jaw-droppers was finding out it's not only bacteria and archaea that use CRISPR systems.
Wait, what? Who else?
Viruses themselves. Some phages have their own CRISPR-Cas system.
Hold on. Viruses are using the antivirus system. Against what? Other viruses?
Sometimes, potentially. But the best studied example is a phage called ICP1, which infects Vibrio cholerae, the cholera bacterium. This phage, ICP1, has its own CRISPR-Cas system. It's related to the bacterial type IF systems.
Okay, a phage with CRISPR. What's its target? Is it attacking the bacterium's main DNA?
Not directly.
Yeah.
This is the really clever, sneaky part. The phage's CRISPR system targets a bacterial defense mechanism.
It targets the bacterium's defenses.
Yes. Vibrio cholerae has these mobile genetic elements called PICIs, phage-inducible chromosomal islands. These PICIs normally get activated when a phage attacks, and they interfere with the phage's ability to replicate. They're an anti-phage defense for the bacterium.
Okay.
So the ICP1 phage uses its CRISPR system to find and cut the PICI elements in the bacterium, disabling the bacterium's defense against the phage.
That is diabolical. The phage uses CRISPR to take out the bacterial anti-phage system so it can replicate freely.
Exactly. It's a counter defense.
I love
A
it.
whole new layer in the evolutionary arms race. And these phage CRISPR systems can acquire new spacers too, adapting to changes in the bacterial PICI elements. It's an ongoing battle.
Mind blown. Are there other examples?
Yeah, there's evidence of CRISPR systems in some archaeoviruses too. Some seem to have mini CRISPR arrays that target other viruses or maybe plasmids that might be competing with them inside the host cell. So yeah, potential interviral warfare using CRISPR.
Fighting viruses with CRISPR, the complexity is just staggering. This whole journey from, noticing weird repeats, figuring out the immune function, the molecular details, the evolutionary battles, even viruses using it, it all loops back to the application everyone knows about now, gene editing.
It does. That's where the story exploded into public consciousness. One scientist, particularly Jennifer Doudna and Emmanuel Charpentier, figured out the key parts of the type 2 system, the Cas9 protein, the cRNA, and that trach RNA. They realized how it worked.
Yeah.
Cas9 is like programmable scissors, and the RNA components act as the guide, telling it precisely where to cut the DNA.
So the big insight was, if we can make our own guide RNA, we can send Cas9 to basically any DNA sequence we want in any organism.
That was the revolutionary leap. You design and synthesize a small guide RNA, often combining the cRNA and trach RNA into one single guide RNA, or sgRNA, that matches the specific DNA sequence you want to target in a cell's genome.
Okay.
You introduce that guide RNA along with the Cas9 protein into the cells. The guide RNA leads Cas9 to the right spot in the vast genome. Cas9 makes a clean cut, usually double-strand break in the DNA.
And then what? The cut itself doesn't edit the gene, does it?
No, the cut is just the start. The magic happens next because the cell hates having broken DNA. It immediately tries to repair the break using its own natural DNA repair machinery.
Ah, so you hijack the cell's repair process.
Exactly. There are two main ways. One repair pathway is a bit error-prone and often introduces small insertions or deletions right at the cut site. This usually scrambles the gene's code, effectively knocking out or disabling the gene.
Okay, so you can break genes easily. What about fixing them or changing them?
That's the other pathway. If you provide the cell with a template, a piece of DNA containing the sequence change you want to make along with the Cas9 and guide RNA, the cell can use that template during the repair process. It essentially copies the information from your template into its own genome at the site of the break. This allows for precise edits, correcting a mutation, inserting a new sequence, changing a gene's function.
Find, cut, and then replace or modify using a template. That sounds incredibly powerful.
It is. The potential applications are just enormous, and many are already happening. It's completely transformed basic biological research. Understanding what genes do is so much faster now.
And in medicine?
Huge potential. Clinical trials are underway for treating genetic disorders like sickle cell anemia and beta thalassemia by editing patients' blood stem cells.
Yeah.
There's immense interest in using it for cancer immunotherapy, engineering immune cells to better attack tumors. And as we mentioned, diagnostics using systems like Cas13.
Right, the collateral cleavage diagnostics. What about beyond medicine?
Agriculture is another massive area. Engineering crops for better yield, drought resistance, nutritional value, developing new biotech products. It's touching almost every area of life sciences.
In this specific work, figuring out how to program Cas9 for gene editing, that's what led to the Nobel Prize in 2020 for Doudna and Charpentier.
Yes. Their foundational work demonstrating how the cRNA and trach RNA guide Cas9 and how the system could be repurposed and simplified into a programmable gene editing tool was absolutely pivotal. It unlocked the whole field.
It really is amazing. Tracing this path from a weird observation in bacteria to understanding this complex immune system to a technology that literally lets us rewrite the code of life. It's almost unbelievable.
It is. But of course, like any technology this powerful, especially one that can alter genomes, it comes with significant responsibilities and ethical questions, especially when you talk about making changes in humans that could be passed down generations, germline editing. There are ongoing crucial discussions about how to use this technology wisely and ethically.
Absolutely. That's a whole other deep dive, maybe. So wrapping this up, what's the big takeaway for you, our listener, from this incredible story?
I think it's a powerful reminder of where discovery comes from. Understanding the fundamental, sometimes seemingly obscure biology of the world around us, even this ancient molecular warfare between microbes, can unexpectedly provide us with tools that change everything.
From a bacterial defense against viruses, shaped by eons of evolution, to a precision tool giving us unprecedented power over biology itself.
Exactly. And maybe the thought to leave you with is this. As we get better at wielding this fundamental biological force, this ability to understand and even direct life's building blocks, what new challenges, what new opportunities, what future battles might emerge from that power?